My name is Donnie Walker. I'm an assistant professor in the Department of Biology at Middle Tennessee State University. We are, as a group, we're capturing fundamental and foundational scientific knowledge about bacterial fungal interactions in herptile gut microbiome systems.
We wake up, we get ready, we go out into the field, we collect at a couple different sites. We try and get different species depending on where we are that help to fill in the cracks of the data set.
Well, we've been catching our amphibians and reptiles across the state of Tennessee. And the reason that we're doing that is because we're interested in the fungus Basidiobolus, which lives in the gut of these organisms. And we believe that Basidiobolus has a broad diversity that is currently undocumented, and also that this fungus has broad implications for the structure of the microbial communities that live in the gut of these organisms.
The lab work for this project involves capturing the animals, bringing them back to the setup lab that we have here on site, and we wash the animals with distilled water so that we can get rid of any extra material they might have on them, and maybe some of the microbes that are on the outside can be washed off. And then we put the animals in a moist chamber, which is just a paper towel inside a little dish, and we let them sit there for about 18 hours or so, maybe up to 24 hours, and we're waiting to see if they leave us a fecal pellet. And with that, we'll capture that pellet and we'll process that in the lab. Take that pellet and we vortex it in some water. We can take small volumes of that mixture and aliquot it into separate tubes for the different downstream processing.
We split that 1 milliliter of fecal pellet dissolved in water into 4 parts. We take 250 microliters for making a spore shoot plate, hoping that we can get Basidiobolus to shoot spores on a very thin layer of agar in a petri dish, and so we just gently spot that solution around the plate.
So I've been isolating the fungus that's one of the foci of this project, and so I'm setting up cultures, waiting for those spores to shoot to the agar surface. Then I pick those off using sterile technique and establish axenic cultures, so it's only that one species that we're interested in.
So the capture and release process is really important. When we capture an animal, we're sure to mark the particular site with a flag so that when we release the animals back unharmed and very minimally invasive, using very minimally invasive processes, we're sure that these animals get back to the exact log of capture so that they can continue to live happily and survive and maintain these really important populations of reptiles and amphibians.
How important is outreach to expand scientific research?
Actually, I think that I didn't understand that until this project. I think that as scientists, we are pretty much focused on just publish and communicate our findings to the scientific community. So we usually write papers or little notes, but focus on that scientific community. But outreach with other people, they are not that in touch with the scientific part of everything, letting them know what are we doing and why are we doing it.
You know, it's just an incredible project. It's an incredible group of people that we're working with. A beautiful part of the country, as you can see here. And it has such a sense of discovery. As you can imagine, there are not a lot of people that have studied the herptile gut microbiome and the fungi and bacteria that live in it. So it's just a sense of discovery where we discover something new all the time, and it provides a really high level of excitement. Do it.
What's your call there? Copy, Ghost Rider? Elephant landing?